Welcome to all of you that have joined us from various time zones around the world, and thank you for attending our webinar entitled Bridging the Distance to Large-Scale GMP Manufacturing of Viral Vectors. My name is Andrew Laskowski. I'm the product manager for the iCELLis bioreactor family, and I will serve as your moderator for today's webinar. I'm also delighted to introduce our guest speaker for today's webinar. Margarita Neri is the Director of Vector Process Development at AGC Biologics. She joined AGC, formerly MolMed, in 2012, working in the Quality Control Unit and then in the Development Unit, where she gained knowledge and expertise in the design of vector product production processes. She is involved in process development and large-scale GMP production of lentivirus and AAV. She holds a degree in pharmaceutical biotechnology, and despite being partially Italian, I'm going to butcher this pronunciation, University degli Studi in Milano. She has a PhD in molecular medicine with a study on gene therapy for lysosomal storage disorders. Our webinar today will be divided into two parts, followed by a Q&A session. To start us off, I will briefly introduce the iCELLis bioreactor and describe some of the recent improvements that have been made to the product. Then we will be joined by Margarita for the second section of the webinar. She will share with you some of the recent successes her team has had developing a process on the iCELLis Nano and scaling it up to the iCELLis 500. Now, let's start the webinar. As I already said, today we'll be discussing how the iCELLis bioreactor can be used to overcome challenges associated with scaling up a viral vector manufacturing process. One of the most significant challenges was articulated by J.C. Vanderloo in the Journal of Human Molecular Genetics back in 2016. He writes that an estimated 1 to 2 orders of magnitude increase in manufacturing capacity is predicted to be needed to meet the increasing number of disease indications that may be targeted by gene therapy. And this challenge can be overcome using one of two strategies. The first strategy is more of a brute force strategy. In this approach, the industry chooses to invest in additional manufacturing capacity by building more facilities. The second strategy, and arguably the more desirable of the two strategies, would be for new technologies to emerge that allow existing processes to become more efficient, thereby reducing the manufacturing footprint and allowing us to better utilize existing facilities and space. Using the iCELLis bioreactor helps to achieve this, or helps us to overcome this challenge by adopting that second strategy. So companies that have used iCELLis have noticed that they can reduce their manufacturing footprint in comparison to two-dimensional cell culture technologies like flatware. And in addition to that, they can adapt their process to the iCELLis with minimal process development required. And what that allows them to do is avoid time-consuming process development steps, such as developing a suspension cell line to use in a suspension bioreactor. For these reasons, the iCELLis has had a lot of commercial success. It's currently used in the manufacture of 6 approved commercially available human therapies, and that includes 4 in vivo gene therapies. For those of you who are not as familiar with the iCELLis bioreactor, it's what we call a fixed bed bioreactor. And a fixed bed bioreactor is simply a bioreactor that has been optimized for the growth of adherent cells. As you all know, adherent cells are unique in the sense that they need to be attached to a surface, or what we sometimes call a substrate, in order to proliferate and grow. In the iCELLis bioreactor, we call that substrate the fixed bed. And the fixed bed is comprised of a material that we refer to as carrier strips. Carrier strips are pictured on the image on the far left-hand side of this slide. And what you can tell by looking at the image is that, at least at the macroscopic scale, these carrier strips resemble pieces of confetti paper. Although unlike confetti paper, they are cut and folded to very precise dimensions. We take these carrier strips and we compact or compress them into a structure that we call the fixed bed. A picture of the fixed bed is shown second from the left here on this slide. This is a computer-generated image of a fixed bed for an iCELLis 500 bioreactor. You can see that the shape resembles the shape of a donut. And in fact, several of our customers oftentimes refer to our iCELLis 500 vessels as donuts. While these carrier strips resemble pieces of confetti paper at the macroscopic scale, it can be a bit misleading. If you put these under a microscope, what you'll see is that these carrier strips are actually consisting of a series of non-woven fibers. And when you have a cell suspension and you're running a batch and you pass that cell suspension through the iCELLis fixed bed, what happens is that the cells are initially physically trapped at the intersection of these fibers. Over a short period of time, usually 1 to 2 hours, the cells will form a physical adherence point with the fibers. And over time, they'll begin to proliferate and grow along the length of the fibers, eventually filling in the interstitial spaces between the fibers, as pictured here on the far right-hand side of this slide. Fixed bed bioreactors like the iCELLis have several benefits compared to traditional two-dimensional flatware. The biggest benefit is the scalability. Unlike flatware, where you're only able to scale out your process and thereby consuming a large footprint in your manufacturing facility, the iCELLis bioreactor can be scaled up. The smallest size iCELLis comes in a size of 0.5 meters squared, and the largest size iCELLis has 500 total meters squared, meaning that you can scale up your process 1,000-fold before there's any need to scale your process out. The iCELLis also offers you several opportunities to reduce the cost of your process. Going back to a couple slides ago, I mentioned that you can reduce your footprint using iCELLis. And the reason for that is because you reduce the need for ancillary pieces of equipment typically associated with the 2D flatware process, such as incubators and biological safety cabinets. Eliminating these ancillary pieces of equipment not only reduces the footprint of the process, but it also reduces the capital expenses and operational costs associated with these pieces of equipment. Unlike traditional flatware, which typically requires a small army of operators to operate the dozen or even hundreds of flasks, the iCELLis requires only a couple of operators per vessel. And this allows you to reduce your number of operators and therefore reduce your overall labor cost. And finally, the iCELLis provides a higher degree of process assurance. It comes integrated with single-use pH, DO, and temperature sensors, which allows you to better control the cellular environment, leading to better cell growth and higher productivity. The iCELLis is also a closed system. There's no open handling associated with it, which reduces the risk of contamination. And finally, the iCELLis is fully single-use, and so there's no cleaning validation required. All of these benefits will ultimately culminate in your ability to reach the market faster with your drug. And as many of you know, in the world of gene therapy, there is no prize for getting to market in second place. You want to be the first and the fastest to commercialize your drug. There are also a few benefits of the iCELLis compared to other fixed bed bioreactors. I mentioned before that the iCELLis is the most widely adopted fixed bed bioreactor technology in the market. It's used for 6 commercially approved human therapies, and of the 7 or so commercially approved in vivo gene therapies, 4 are produced on iCELLis, as well as many others that are currently in the clinical trial pipeline. Hopefully this helps to illustrate how instrumental the iCELLis is in enabling a viral vector manufacturing process. The iCELLis also has really nice automation. The software, the control software, is 21 CFR Part 11 compliant, and there are other features of the automation that make the iCELLis really easy to integrate into a GMP manufacturing environment. The iCELLis bioreactor is also supported by a truly global company in Cytiva. We have secure global supply chains, but we also provide you with personalized, localized support. And that support comes in the form of a local sales team, local technical and scientific experts that will help you develop your process and train you on the instrument, as well as local service support for installation and maintenance services. And finally, the iCELLis has more advanced process analytical technologies compared to other fixed bed bioreactors. As I mentioned a few minutes ago, we integrate a biomass sensor with the iCELLis. We're the only fixed bed bioreactor to have a biomass sensor integrated. And the biomass sensor allows for continuous inline measurement of your cell density. And in some cases, it may even allow you to measure the concentration of your product. There are 2 iCELLis systems available. The small scale or bench scale system pictured on the left-hand side of the slide is called the iCELLis Nano. And although this bioreactor is most commonly used for process development, it can also be used for small batch GMP manufacturing. It has a maximum surface area of up to 4 meters squared. On the right-hand side of the slide is what we call the iCELLis 500 Plus bioreactor. It's our large-scale or commercial-scale system that is most commonly used for GMP manufacturing, and it has a maximum surface area of up to 500 meters squared. This next slide summarizes some of the case studies that are published in the iCELLis bioreactor. These 5 particular case studies are available in the peer-reviewed literature, but there are over 100 different iCELLis case studies that can be made available in the form of posters or conference presentations or journal articles or application notes. So if you don't see your application on this slide, or if you would like to see more information about any of these listed applications, please reach out to us and we'd be happy to provide you with additional case study information. Now I'd like to pivot for a moment and talk about a recent improvement that has been made to the iCELLis, and that improvement is the launch of our 2nd generation control software, which we call EmpathLink software. And this is one of my favorite improvements that we've released for iCELLis, and that's because as a former process development scientist myself, I have been in the past intimidated by very difficult to use bioreactor control software. And I've always wondered why this is the case. Like many of you, I will oftentimes get a new iPhone every several years. And when I get a new iPhone, I'm never intimidated by my inability to learn the new operating system or the new software that's on the device. In fact, like many of you, I look forward to my new operating system because it's generally easier to use and has more features than the previous system. Apple's not sending an army of technicians around the world to teach their customers how to use their operating systems because it's just naturally intuitive. And the truth is that bioreactor control software can be the same way. It's just that no one's taken the time to make a very user-friendly interface until now. So EmpathLink software has undergone over 100 hours of usability testing. And as a result, it's an extremely simple, it's a streamlined, easy to use, intuitive user interface. If you have reservations about trying the iCELLis reactor because you're intimidated to learn a new software, then rest assured that that will not be the case with this technology. In fact, what you'll find is that the software contains a lot of really easy to use features that will help you better understand and optimize your process in the iCELLis reactor. Now, unlike more traditional benchtop bioreactors, the control software for iCELLis is not installed on the reactor's control tower. Instead, we install it on a separate device that we call the EmpathLink server. And there's 2 reasons for this. The first reason is that the EmpathLink server can act as a networking device. You can network multiple iCELLis bioreactors together with the server, as well as multiple human-machine interfaces, which gives you and your colleagues the convenience of accessing any iCELLis bioreactor from any interface that's on that network. The second advantage is that this scheme allows you to basically operate as many iCELLis bioreactors as you would like, but only requiring you to purchase 1 software license. So in a situation where you want to scale out your process development lab, let's say you have 6 iCELLis bioreactors and you want to increase it to 12, you simply have to buy 6 new bioreactors. You don't have to also buy 6 new software licenses. And so that makes it more economical to expand your lab. The EmpathLink software also has a few exciting new features. So one of those features is a mobile phone application that you can download and install on your smartphone. This allows you to monitor and even control your bioreactor essentially from anywhere. We've coupled this new feature with a notification feature that will allow you to also receive email or text message alerts if your bioreactor were, for example, to encounter a high pH alarm. So if you're not at work, say you're just relaxing at home or you're out to lunch or out to dinner, and something goes wrong with your reactor, let's say you get that high pH alarm, an alert will be sent to your phone. You can then read that alert and log into the software application on your phone, monitor the reactor and make the necessary changes. and have the peace of mind that your process is running smoothly without having to go into lab and disrupting your day. A few other new features include an improved recipe engine. Users now have the ability to design multi-phase recipes, such as a recipe with a phase for startup, a phase for cell expansion, and a phase for product production, and even specify user transition points between those phases. We've also improved our data trending interface, including adding the ability to overlay trends from multiple batches on one trend graph. And finally, we've embedded training videos throughout the software. So if you, for example, haven't run the iCELLis reactor in a while, or there's a new colleague in your group, or perhaps you've just forgotten the best practice associated with an operation like pH probe calibration, you can click on one of those links. It'll bring you to our training video platform, and you can watch the video on pH probe calibration to ensure that you perform that operation correctly. The last topic I want to comment on before I turn things over to our guest speaker, Margarita, is I want to revisit the topic of biomass sensors. So as I mentioned a few slides ago, the iCELLis bioreactor is the only fixed bed bioreactor to integrate a biomass sensor. And biomass sensors can be used to measure your cell density, as well as in some cases, even measure your product concentration. And the reason we've integrated a biomass sensor with iCELLis is because, as many of you know, measuring cell density in fixed bed bioreactors or any type of adherent system can be challenging. The traditional methods associated with cell counting have some pretty major drawbacks. So one of those methods is performing a nuclei count. But some of the drawbacks associated with that method are, number one, you have to open the vessel and risk potential contamination in doing so. Nuclei count measurements are not continuous. They're performed discreetly when the operator chooses to do so. And they're not scalable because you cannot open the larger scale reactor to perform said nuclei counts. Another method that's used commonly is metabolite analysis. So typically with this method, you'll measure glucose or lactate concentration and use that as a surrogate indication of cell growth. But glucose and lactate do not directly correlate with cell density. And so oftentimes this method is imprecise or inaccurate. And that's why we have the biomass probe, which gives us somewhat of the best case scenario of both of these different techniques. So if you have an integrated biomass sensor like the iCELLis does, there's no open handling required to get a cell density measurement. The biomass sensor provides these measurements continuously, and it does so in a scalable manner. It's available on both the Nano and the iCELLis 500. So the biomass probe can be used as a process development tool in the Nano to better understand, characterize, and optimize your process. And then when you scale up to the iCELLis 500 and you're in production, it can be a tool that helps you ensure process consistency from batch to batch. And now, I'm delighted to introduce Margarita to begin the second phase of this webinar. Margarita, please take it away.
Thank you, Andrew. First of all, I would like to present you AGC Biologics. AGC Biologics is a global CDMO that offers end-to-end capabilities. We have industry-leading expertise in process development and in internalizing client processes. We have experience with lentiviral vector, retroviral vector, and AAV manufacturing, and also in cell therapy products. We do the customization, development, qualification, and validation activities for manufacturing processes and analytics. We have authorized GMP facilities for clinical and commercial supply. We also have a very complex quality management system in line with the European GMP requirement and FDA guidelines. We also have internal QPs for the release of the products. The majority of the analytics are performed in-house in order to reduce the overall turnaround time. These are the three AGC sites for cell and gene therapy. Milano is the AGC facility where all the vector platforms have been developed. In Milano, we are about 300 employees, and we perform manufacturing of viral vector and cell therapy products to cover from preclinical phase to market. We have 5 streams of GMP suites for vector production at different scale, from 50 liters to 200 liters. More recently, AGC acquired a facility in Colorado for manufacturing of viral vector and cell therapy, where we have just concluded the process transfer from Italy. Moreover, in Heidelberg, we have a facility for mRNA and plasmids manufacturing. In order to answer to the demands of the cell and gene therapy fields in terms of productivity, flexibility, and robustness, we recently launched our brand new vector platforms, Pronto LVV and Bravo AV. These platforms include vector manufacturing processes, both for adherent cells and suspension cells at different scale. They have been developed in Italy, in Milano, and the names reflect the main features of these platforms: quality, speed, and readiness, not only to reach the GMP manufacturing, but also more advanced phases, because some of them have been already approved by authorities. The vector production platforms that we offer include not only the process itself, but also the starting materials. We have already released a master cell bank available for our customer. We have packaging plasmids and also the backbone of the transfer plasmid to be shared with our clients in order to clone inside their gene of interest. Moreover, we offer the process protocols and a very complex panel of analytical methods. As I mentioned before, we have developed both suspension and adherent processes. And today I will focus my presentation on the adherent platforms, and in particular on the use of the iCELLis bioreactor system. We established both LVV and AV processes from 50 liters to 750 liters using HEC2net 3T master cell bank or HEC2net 3 in transient transfection. And all processes are ready to use and allowed to move from the gene of interest through process establishment, process scale-up, and GMP manufacturing in less than 1 year. This is the flowchart of our historical process in cell factoring for the production of lentiviral vector. This process is our gold standard for lentiviral vector and is now used for different products also on the market. The process is based on 24 cell factory 10 trays and is performed by cell towing and expansion, transfection with plasmids and PI. We perform harvest of the biovector, clarification and prefiltration, addition of benzonase, and then we have the load on the anionic exchange chromatography for vector purification, a step of concentration with oligofibers, and a step of gel filtration for the size exclusion chromatography in order to further purify the vector and also to resuspend the vector in the final buffer. In the end, sterile filtration and filling. We were able to obtain a very good infectious viral titer. Here reported all the data that we obtained in our GMP batches. The average of the titer in the biosupernatant is about 10 to the 7 TU per ml, and in the final filling vector we are about 10 to the 8 TU per ml. Since a customer that performed the first clinical phase with the cell factory process started asking for a scale-up with reduced comparability effort, we decided to start working with the iCELLis platform, that is a packed bed bioreactor available at different scale where cells can grow easily in adhesion. We developed a process for lentiviral vector based on the 133 square meter bioreactor at full scale. And here is reported the flowchart of the process. Cells are towed, expanded in the bioreactor, is performed DNA transfection with the PI inside the bioreactor. We perform an harvest in perfusion. Then we add benzonase to the bulk vector. We perform clarification and prefiltration. And then there is a step of anionic exchange chromatography, of course, for the vector purification, and a step of concentration and then diafiltration with holofiber, in which we also rebuffer the vector in the final medium for the formulation. In the end, of course, sterile filtration and filling. Importantly, we exploited the scaled-down iCELLis Nano to design a scaled-down model representative of the whole process, upstream and downstream, useful for process development, but also for process characterization and also for feasibility studies that we perform for our customer. All the instruments that you use in the full scale are single-use instruments, included the ActaReady system for the chromatography and the ActaReady Flux for the managing of the concentration and diafiltration step. In this graph are represented all the runs in the 1.06 square meters iCELLis Nano bioreactors that we perform during the process development. Blue and green dots represent physical and infectious viral titers obtained with the GFP transgene, that is the transgene that we use for the vector for the process development. While in yellow and in red, the data obtained with other gene of interest from different customers. Working on several parameters as seeding density, amount of DNA, growth condition, we were able to scale up the process in the full-scale iCELLis 500 133 square meter bioreactor. Here is reported the infectious viral titer of bulk supernatant and in the final filled vector obtained with the scale-down process in iCELLis Nano and in the full-scale process in iCELLis 500. We were able to scale up in iCELLis 500, confirming the full scalability of the system and of the whole process. The productivity have been confirmed with different starting materials, different gene of interest, but also different set of packaging cells and packaging plasmids. And the overall process recovery of the whole process, including the downstream and the sterilizing step, is about 20%. We also evaluated the residuals in terms of hostile protein and total DNA of the vector produced with the iCELLis process, showing that the impurities profile of vector produced in bioreactor was comparable and even better to our gold standard process in cell factories. And we also tested the functionality of vectors transducing the target cells mainly used for cell and gene therapy, like T cells and CD34 cells, obtaining comparable vector copy number and transduction efficiency level for the vector produced in bioreactor versus the cell factory process. And of notice, the iCELLis process is characterized by a very low residuals level, therefore could particularly indicated for lentiviral vector in vivo gene therapy. Here, there is a summary of the productivity in terms of infectious titer of our 200-liter process in iCELLis. I reported here the average data of each full-scale run with different transgene. We are now working to further increase the packet bed area and also to test affinity chromatography to further increase the vector purity. And while the 200-liter process with the 133 square meters packet bed is already running in GMP, we are carrying on the development with the aim to increase the packet bed size and consequently the bulk vector harvest and the batch size. Here are shown some preliminary results that we obtained with the 2.66 65 square meters and 4 square meters iCELLis Nano, in which we demonstrated that we obtain comparable titers in terms of P24 and TUML, increasing also the culture area, meaning comparable productivity per square centimeter. The total harvest from the full scale in 500 square meters will be 750 liters, that is highly competitive with the more known suspension systems. Moving now to the AV manufacturing platform. For AV, we choose to use the iCELLis 500 333 square meters using heteronite 3 cells, and here is depicted the flow chart of the process. Also in this case, we have cell towing and expansion in the bioreactor, DNA transfection, and then we have a step of lysis and benzonase treatment. And in this case, we have to differentiate if we are producing a released serotype or a retained serotype. In the case of release serotype, we collect the whole cell supernatant and also the cell lysate. But if we are producing a completely retained serotype, we just collect the cell lysate. And in this case, we perform clarification, and if the serotype is released, we perform a first step of concentration by hollow fiber in order to reduce the volume, because for release serotype, we collect 480 liters of bulk vector, while if we lyse the cells and we collect only the cell lysate, we collect about 70 liters. Then there is a step of affinity chromatography for vector purification, a step of polishing with anionic exchange chromatography with the aim to enrich in full versus total particles. Then there is a step of concentration and again diafiltration for the resuspension in the formulation buffer, sterile filtration and filling. And also in this case, we have a scaled down model of the process based on the use of the iCELLis Nano that accordingly to the secreted or retained serotype starts with 8 liters or 3 liters of bulk vector. In this graph are represented all the runs in iCELLis Nano bioreactors that we performed during the development of the process for the manufacturing of AV6, that is a retained serotype. Using different packet bed size and also testing different cell line, we were able to reach in the bulk vector more than 10^11 vector genome per ml. This upstream process has been then scaled up in iCELLis 500 333 square meters with AV6. Moreover, we tested our process also for the production of AV8 and AV9, and we demonstrated the robustness and the high productivity even with the released serotype. And also we scaled up in the iCELLis 500 a process for the manufacturing of AV8. In this slide are shown the productivity data in terms of vector particle per ml, the dots, and vector genome per ml, the bars, of the full-scale run for AV6, that were 70 liters of bulk vector, and AV8, 480 liters. And you can see data pre- and post-clarification of the full-scale processes compared with the data obtained in iCELLis Nano, the light blue and light green. And this also again confirmed the good scalability of the platform from the iCELLis Nano to the iCELLis 500, and also an efficient recovery of the clarification step. Moving to the downstream, in this graph are depicted the total vector genome and total vector particles across the different downstream steps. And in this case, the total process recovery is about 30%, and it's possible also to appreciate the enrichment in full particles obtained from the bulk vector to the final fillet product that are reported on the bars. We evaluated also the residual levels, also in this case, in terms of hostile proteins and total DNA of the bulk vector of full-scale runs versus the run performed in Icelis Nano, demonstrating also for this aspect the comparability of the two systems. Of notice, residuals like total DNA and hostile protein are undetectable in the final filled vector already, in reality, already after the first affinity step. So here are the productivity data for AV platform at a glance. With the AV6, we produced 1.5 10 to the 11 total vector genome per batch, while with AV8, 1 10 to the 16. As already mentioned, also for a lentiviral vector, also for AV, we are now working on the further increase of the batch size. And we demonstrated at first the comparability in terms of productivity with the low compaction and the high compaction. And then we move to test in the bigger bed size, 4 square meters, and we obtain also in this case a comparable vector genome per square centimeter. And so we demonstrated that also AAV can be manufactured with the adherent system, obtaining till 750 liters of bulk vector. In conclusion, Icelis bioreactor system allow the development of fully GMP compliant manufacturing process. LVV and AV processes, in addition, are highly productive and with a good vector quality in terms of infectivity and the levels of impurities. And the Icelis-based process are robust and reproducible and allow to reach till 750 liters of bulk vector. And we also demonstrated that Icelis Nano is an ideal scaled-down model for process development and also for process characterization. The batch size, of course, produced in bioreactor is cost-effective in terms of number of patients treated versus the cost of the production and of the QC. I would like to thank you for your attention, and now I leave the word to for the questions. Thank you.
Thank you, Margherita. I really appreciate you sharing, and congratulations on the success that you've had scaling up your process to the SLS 500. We will now move on to the Q&A session. And let's go ahead and get started. So the first question that I have here, I think, is intended to you, Margherita. Someone wants to know what's the amount of time to go from development to GMP for an AAV process?
Yes, thank you, Andrew, for the questions. So yes, we claim about 1 year starting from the starting of the contract to the GMP manufacturing. Usually, what we offer to our clients, both for AV and LVV, relying on our platform processes, is a feasibility study in which we take advantage of our scaled-down models, and we can offer the clients to test their transgene in our platform with scaled-down models, so with also a few efforts in terms of materials and economics. And then we produce a vector in a development that we can send to our customer to be tested in their system with their cell target cells. And after the verification of the quality of this vector, we scale up to the full scale. We perform a full scale run in the development, or we directly perform an engineering run in the GMP according to clients' requirements, in order also to accommodate stability studies or other studies that are required, for example, preclinical studies or toxicity studies with the vectors produced in the development space. And then we move to the GMP.
Okay, thank you. This next question, I think, is probably directed to me. There's actually a couple of different questions around the biomass probe. So I think perhaps all these questions just be best summarized in terms of how does the biomass probe work? And I think that's a really good question. So the principle is called radio frequency impedance. Essentially, the way these probes are built is there are two electrodes that generate a very weak electric field. There's a positive side to that electric field and a negative side. And the poles of that electric field switch. And as the speed at which they switch, that's called the frequency. And when those poles switch, charged ions move back and forth in solution at really, really fast rates. And cell membranes and other biological structures will impede the movement of those charges. And what you'll have is, for very small periods of time, you'll have positive charges aligned on one side of a cell membrane and negative charges aligned on another side of the cell membrane. And when that happens, the membrane acts as a tiny little capacitor, and the biomass sensor can read the strength of that capacitance from the cell membranes. And the strength of that capacitance is proportional to the cell density. Next question here are, what are the pros and cons of transient transfection versus a stable producer cell line? Margarita, why don't I pass that question off to you?
Yes, thank you. Yes, there are some pros and cons for each of these two systems. We mainly work with transient transfection because it's mainly more flexible. You can change the transgene and the packaging plasmids very easily. And in particular, if we are dealing with clinical trials, phase 1, 2 is more indicated transient transfection, because when you have to establish a stable cell line for a stable production, usually this is done for the more advanced phase of the development, of the clinical development of the product, so phase 3 and commercial phase. From our experience, the stable cell line usually are a bit less productive compared to the transient transfection, but of course allow to perform a harvesting perfusion and probably to harvest the vector for a time windows that is higher, so it's a balance. There are also systems that are, let's say, like an hybrid, that's semi-stable, so you have stable integrated in the packaging cell line, the packaging plasmids, and you have to transfect only the plasmid, and this could be a good, let's say, balance because allow to have the flexibility and not to invest so much resources in the manufacturing of the GMP plasmids. But also in this case, we have to work a bit to increase the productivity of this kind of systems.
Okay, thanks. We have a few more questions coming in here. A lot of these questions actually have to do with comparing adherent and suspension processes. Let's hold off on those questions just for a moment, though. There's a question about how do we ensure that there's homogeneous media in both the bioreactor and in the recirculation media tank. And I'll take make an attempt at this question first, and Marguerite, if you have any other comments, you can answer as well. So one of the things that we did a couple of years ago is we created a glucose concentration simulator that uses the pump speeds as an input to basically not simulate, but calculate what the theoretical concentration of glucose would be in the bioreactor versus the mixing tank. And what this spreadsheet allowed you to do was optimize the recirculation rate. We call it the recirculation rate, the speed that media is pumped out of the vessel and into the recirculation bag, and then from the recirculation bag and back into the vessel. And it's not possible to pick a recirculation speed so that the glucose concentration would be identical. But it is possible to pick a recirculation speed so that the glucose would be, let's say, plus or minus 5% difference. And so that would be the recirculation speed that we'd recommend to our customers to ensure that the media was homogeneous between the vessel and the tank. Margarita, do you have anything you'd like to add to that?
just to confirm that we apply this calculation and this way of work for our processes when we need the recirculation, and it works.
Okay. Yep. All right. There's a question here. I think the person asking the question referenced your lentivirus process at the 200 liter scale, and that the final volume at the end of that process was 2.5 liters. And they're wondering, was anything lower tried, maybe something in the range of 1 to 2 liters? And if so, was recovery and purity similar?
Yes, so the final volume and the final concentration of the vector is something that we agree with our customer. Let's say the 2.5 liter is our proposal, is our, let's say, standard final volume, but of course it's something that we can agree. These 2.5 liters is mainly due to the, let's say, dead volume, to the, let's say, dead working volume of the equipment that we use for the final concentration at a larger scale, that is the acta ready flux. If we want to go further concentrate till 1 liter, for example, we need to move to a second concentration step with a different equipment that allow us to go lower with the concentration factor, and as let's say, less dead volume in manifold and fibers. We did some trials in order to accommodate this second concentration during the process development, and the recovery is similar because we do not lose so much vector during the concentration and diafiltration steps. The yield is about 80–90%, and also in terms of purity, the purity is the same, because we at that time of the process we already performed the purification with the chromatography, and so the impurity level is already very, very low, and so adding a further step we do not alter this profile.
Okay, great. Now let's get into some of these adherent and suspension questions. So there's a question here. Can you comment on whether you have compared specific productivity and impurities in the same product in HEK 293 adherent versus suspension cells? And if so, which approach was most successful?
Yes, I can comment about this point. Of course, adherent and suspension system are very different in terms of impurities profiles of residuals, in terms of hostile proteins, and for example, total DNA released both by the, let's say, mainly released by the producer cells. When we perform adherent process, when we collect the vector, we do not have let's say the cells remains attached to the substrate. We just have some cell debris, and we perform a clarification, and the levels of impurity is very low already at the beginning, in particular with the isolysis process for lentiviral vector, because the packet bed, it's like a filter itself when we collect the supernatant. While for the suspension system, if we use a depth filtration, we have also damage of cells, because the whole cell suspension is passed through the filters. So we have cell damage and the release of cell proteins and cell DNA. And so the impurities levels, let's say at the end of the upstream phase in the bulk vector is consistently higher in the suspension harvest compared to the adherent system. The good news is that with our downstream process, we are able to reach at the end of the process, so in the final field vector, comparable levels of impurities that are mainly under the limit of detection of our analytical methods. So even if the starting material has different levels of impurities, in the end, we have, let's say, a vector quality that is very comparable with the two systems.
Okay, and I think related to that question, someone was wondering why you're working in parallel on adherent and suspension platforms.
Yes, so we are a CDMO, so what we decide to do is to be aligned with the market requests. In this moment, clients request both a suspension and adherent system, so we are ready to offer both of them. Of course, there are pros and cons of these two systems. For the adherent system, for sure, the pros is the higher purity, but also the higher stability of the vector. The cons is the presence, of course, of the FBS in the growth phase of the producer cells. And so even if we demonstrate that at the end of the downstream we have undetectable levels of residual BSA, but we still have this component in the manufacturing process. And the suspension is highly appealing because, let's say, is more related to the traditional biotech production, proteins and antibodies, and is a system that is more scalable till 1,000 liters, 2,000 liters and more. So these are mainly the two aspects that client, and depending on clients, on their, let's say, preferences, they ask for the suspension or for the adherent system.
Okay. And I think the last question here related to adherent versus suspension, I think I'll attempt to answer this one. There's a question about, is there a preference? Is adherent cells or suspension cells preferable? Which one's preferable? And I think the answer to this question probably depends a lot on your circumstances. I think if you have a high producing suspension adapted cell line, you are much more likely to prefer the suspension platform versus the adherent platform, largely because suspension processes are a little bit more well understood. We have a lot of institutional knowledge from CHO and monoclonal antibodies, knowledge around characterizing processes and scaling them up to very large volumes. But if you don't have a suspension cell line, I think you'd probably prefer to use an adherent platform. And I think there's still, at this point, there's probably a little bit less risk with an adherent platform. You can take whatever cell you're currently using in roller bottles or tea flasks, and you can put them into a technology like the iCELLis with very minimal process development, and you can develop a fairly large scale process in a very short period of time. So that's maybe just a long-winded way of saying it depends. But I will say that from the Cytiva perspective as a technology supplier, we have both adherent enabling technologies as well as suspension enabling technologies. We haven't discussed the AccelerX bioreactor, the legacy AccelerX bioreactor. It wasn't the topic of today's webinar. But if you're a company that's somewhat agnostic about adherent or suspension and you want to come to one place for all technologies, I think we can help you out. Let's see here. There's a good question for you, Margarita, about your downstream process for AAV. Is it able to separate empty from full capsids?
Yes, in our platform, but for the adhesion and the suspension, we add a step of polishing with anionic exchange chromatography. We worked a lot on this step, testing different membranes, monoliths, resins, and in the end, we obtain a good recovery of this step and a good enrichment. We end our processes with about 80%, but even more for some serotypes of full versus total particles.
Okay. And I think we probably only have time for 2 more questions here. And I think these are probably directed at me. So one of the questions is, what is the analytical technology for determining the titer of vectors online within the reactor? So I think this question relates to the biomass sensor that I alluded to earlier in the webinar. And I talked a moment ago about how these biomass sensors work, about how ions move back and forth in the media as the polarity of the electric field oscillates. And the capsid of viruses also exclude the movement of ions, or I should say impede the movement of ions. The difference is that viruses are a lot smaller than cells. And so there needs to be a lot more of them, or there needs to be a larger, the virus have to be larger in size in order for them to, in order for the strength of that capacitance signal to exceed the limit of detection of the biomass sensor. So the biomass sensor is not going to be able to measure virus concentration for every application. Some of the smaller viruses or viral vectors like AAV, for example, or some of the viruses that have very low productivities, you might not be able to, in fact, you probably won't be able to measure their concentration. But if you're interested, there's a paper that was published by Merck, not Merck Millipore, but Merck, the US vaccine company, where they used a biomass sensor in a reactor to measure the concentration of an undisclosed virus. I can, if you're interested, I can follow up with you offline and provide you the citation for that paper, and you can look into a little bit more detail. But we're not claiming you can use the biomass sensor for every viral vector or every virus, but it's possible to use it for some. And then the final question here is, what challenges have you faced in determining KLAs in fixed bed bioreactors? So I've done some KLA work, and I'm aware of ongoing KLA work at Cytiva, so I'll comment on this real briefly. But for those of you that have had some experience with the iCELLis Nano, you'll know that there's a very large surface area relative to the volume of media that's overflowing in that falling film. And because of that, the falling film isn't always consistent from vessel to vessel. In the iCELLis 500, this is not an issue because the volumetric flow rate is much larger relative to the surface area. So you have a fully developed falling film. In the iCELLis Nano, that falling film is a little bit less developed. And because it's less developed and a little bit less consistent from vessel to vessel, you have different surface area between that falling film and the gas. And so obviously if your surface area is a little bit different from vessel to vessel, that's going to have an impact on your KLA from vessel to vessel. So I would say that that's the biggest challenge in characterizing the KLA of these iCELLis nano vessels and nano vessels specifically. Again, this problem is not really an issue for the larger iCELLis 500. And with that, we'd like to conclude today's webinar. Thanks again for spending a part of your day with us, and we hope you enjoy the rest of your day and the rest of your week. Thank you.
Thank you.